Okay, so this is the extra lecture for honors um, about plankton. So just a brief review of a couple things that we're going to talk about in class, and then we'll start talking about viruses and bacteria and fungi. So plankton, there's lots of different types of marine plankton. In class, we're going to talk about phytoplankton and zooplankton, which are the last two that are on this list. But because your honors, you get to talk about viruses, bacteria, archaea, and fungi. But these are all types of marine plankton. So plankton, some typical misconceptions about them is that they're small and that they're animals that are eaten by whales. But the reality is that they are both plants and animals. And the three requirements that you need to have in order to be considered plankton is that you are alive, you drift, and you float. Okay? And there is no mention of size. So it doesn't matter what size you are, you can still be considered to be plankton as long as you are alive, you drift, and you float. So this picture right here, this is a picture of a plankton. Okay, so let's look at viruses. Virology is the study of viruses, and there are actually tons and tons and tons of viruses that live in the ocean. In fact, viruses are probably the most abundant things that live in the ocean, and they participate in like all food webs and are actually extremely important to the ecology of the ocean. However, most scientists don't think that they're actually alive, because if you remember when we talked about what it means to be alive, we talked about the two characteristics that are associated with life, which are respiration and reproduction. And in respiration and reproduction, marine viruses require another living thing in order to be able to do that. So they cannot reproduce or undergo any sort of cellular metabolism at all. They require something else to do that for them. So most scientists don't consider them to be alive. However, they're the most abundant things that live in the sea, and you'll hear that term, the term life and stuff referring to viruses a lot, but most people don't consider them to be alive. So, and they are hugely important in marine food webs, but again, they're not considered to be alive. So, what are viruses? Viruses are made out of little pieces of RNA and DNA, and they're surrounded by a protein coat. And so that protein coat and that RNA and DNA are considered to be what we call the nucleocapsid. And they do not have, again, they don't have any metabolism. They don't have any organelles. They just have that DNA or RNA surrounded by protein. They have to replicate, reproduce within a host cell, and they do infect all groups of living things. So you can get a virus. They can infect humans. They can infect plants. They can infect all kinds of animals. So you can have specialized viruses that will infect certain things, like, for example, bacteriophages are specialized viruses that specifically infect different bacteria. So they can infect a lot of different things. So here's your viral structure. The virus, when it's outside of the host cell, is called a virion. And then when it's inside of the host cell, it's virus. The virion is composed of that nucleic acid core, so that DNA or RNA surrounded by the protein coat. Which the protein coat is called the capsid, and the two of them together is called the nucleocapsid. And viruses may have what we call an envelope, which is a membrane that comes from the host cell's cell membrane. So you can have like your little DNA or RNA. Okay, so here's our little DNA surrounded by the protein coat. So those two things are called the nucleocapsid, and then surrounding that, you can get a little coating from the host cell's cell membrane, which is called the envelope. So that's what it would look like. Here's a couple pictures to help you see. So this top part, okay, right here, this would be the nucleocapsid. Okay, that's the protein coat that you see right there, and then the DNA or RNA would be located inside of that. You have different shapes of viruses. You can have what are called icosahedral viruses. That's basically the capsid has 20 triangular faces. So it's like a bunch of different protein subunits that are put together, and it gets this icosahedral shape. So that's this top part right here that I circled in red. That would be the icosahedral shape. So if you just had a virus that was just that, that would be icosahedral. A helical virus is made up of a bunch of protein subunits, and the protein subunits spiral around the central core of that nucleic acid. So this bottom part right here that I'm circling in green, that would be like a helical virus. So that would be, if it was just that part, that would be a helical virus. So you can see the little protein subunits that spiral around, and then you'd have the DNA or RNA inside of there. A binal virus is a virus that has the icosahedral head and the helical tail. So the whole thing together that you see in this picture, that would be a binal virus, because it's got the icosahedral head and then the helical tail. You can have some viruses that will have filaments and stuff to help them to attach to their host and to infect it. So for example, bacteriophages, the specialized viruses that infect bacteria, they look like this. They're binal, and then they've also got like little filaments that stick off down here at the bottom that help them to actually attach to their host bacterial cells and then inject their DNA or RNA into them. So you have those different kinds of shapes of viruses that can have these different specialized filaments to help them to attach. Okay, so viral life cycles. Again, viruses are not actually considered to be alive, but we refer to them as having life cycles. You have two different kinds of life cycles that you can have in a virus. They're the lytic and the lysogenic cycle. So basically, they follow the same path, except for one major difference. In the lysogenic cycle, the viral DNA or RNA remains in the host cell for a longer period than in the lytic. So in the lytic cycle, what happens is the virus rapidly infects the host bacteria or host cell, and then the virus immediately takes over that cell and takes all of the organelles of the cell and cause it to start making the proteins and stuff for new viruses. So viruses infect, they take over the cell, make the cell create new viruses, and then when all the new viruses are created, all of the viruses burst out of that cell, rupture the host cell, and kill the host cell. In the lysogenic cycle, the viruses infect the host cell, but rather than immediately taking over the cell, they instead insert their DNA into the host genome, so they're kind of like sleepers. So the DNA gets put into the host DNA, and then that host DNA gets copied and copied and copied as that host cell multiplies. And then eventually, because of some outside trigger, the DNA of the virus that was inserted comes out, and it then takes over the cell and causes it to start creating new viruses. So here's a picture of the two different life cycles. So this first one up here, okay, this would be the lytic cycle. Okay, so you have the virus infecting the host cell, okay, immediately taking over and creating new viruses, and then those new viruses burst out, and you have new viruses to go infect other cells, and this host cell is sad because he's dead. Okay, so the rupture of the cell is called lysis. So the cell, the host cell is lysed. It dies. It explodes. So the lysogenic cycle. So you still start with infection. Okay, so the virus infects the cell, but instead of immediately going to replication and lysis, the host cell, you can see right here, gets that viral DNA inserted into it. And so as the Cell goes on living its life. It replicates that DNA. Okay, so now all of these cells are infected. It has the virus DNA. Some outside trigger triggers it, and then that DNA comes out, and it causes that cell to start replicating the virus, and then the lysis of the host cell occurs. So really the difference is how long the virus spends in the host cell. So, okay, biodiversity and distribution of marine viruses. Marine viruses are hugely abundant. They are the most common type of plankton that you will find in the ocean. So there's millions, billions of them. So we've done some research and we found that there's between 110,000 genotypes in the ocean of just viruses. And you have viruses that live in the water column themselves, and then you also have viruses that will live in the sediment, so in the dirt at the bottom. So they're all over the place. So what they do, viruses infect host cells, and then they actually, because they infect them and then they kill the host cells, the viruses actually help to control the population of bacteria and then other things that we call microbes, which are microscopic things. So they actually help to control the populations of the bacteria and microbes in the plankton communities. Without them, the bacteria and stuff would get the populations would get way too big, and they wouldn't be able to be supported. So the viruses actually help to release those or control those different bacteria and microbe populations. Also, when the cells lyse, so when the viruses explode out of the cells, that actually helps to release nutrients, because all of the nutrients that were in that bacteria cell or that microbe are now put into the water column. And so that helps to release nutrients for other primary producers to be able to use and to create glucose and start off food chains. So the lysis actually helps to do that. So virus populations need to be controlled as well, because if you have too many viruses, then they kill off too many bacteria, and so you need a balance. You don't want too many bacteria or too few. So the viruses help to control the bacterial population, but then you need to control the viral population. And there's different ways that occurs. You can get viruses that get their DNA or RNA altered by the light. So they live in the surface waters, and the amount of sunlight can actually affect the virus's DNA or RNA and change it, which means that those viruses are then rendered useless, and they can't insert their DNA into a host cell. So the intensity of the light can change the viral DNA or RNA and prevent them from infecting things. Viruses can also get absorbed into suspended particles in the ocean. So you have suspended particles of mucus and plankton, and viruses can get absorbed into those particles, and once they're absorbed, they can't infect their host cell, so they can't replicate. So their population gets controlled that way. Other microbes can actually eat viruses and not be affected by them, so microbes can help to control the population of bacteria or viruses as well. And then if the virus can't find its appropriate host cell, then it can't infect it. So it'll be controlled that way. So that's how viruses help to, or their population gets controlled. Okay. So bacteria. Characteristics of bacteria. So bacteria are very simple prokaryotic cells, which means that they don't have any nuclei or membrane-bound organelles. They still have DNA, but it's not wrapped up in a nucleus. And they also have a cell wall on the outside of their cells. So there's like a non-living like protein and sugar combination around their cell membranes, which helps to protect them. Bacteria reproduce asexually by binary fission. We learned about that last cycle, so you can look it up if you don't know what that means. And there's lots of different shapes and sizes of bacteria. There's 3 major kinds of shapes. So it's called Bacillus, which is a rod shape, so it's like long and skinny, okay, which looks like that. A Coccus, which is a spherical shape, so it's round. And then you also have Spirillum, which is like a spiral shape. The Spirillum is not very common in the ocean, so we don't really talk about it. But the Bacillus, the rod shape, is probably one of the most common. So different types of bacteria. We will group bacteria into different groups based on its nutritional type, meaning how does it get its nutrition, its glucose. So you have cyanobacteria, which are photosynthetic bacteria, which do photosynthesis. And they're found in places where there's lots of oxygen, and they're actually one of the contributors to that oxygen supply because they do photosynthesis. So one of the byproducts of photosynthesis is oxygen. So they help to create oxygen. They store any excess photosynthetic products that they create, like any excess sugar, as cyanophycean starches and also oils. So you don't really need to know that, but that's how they store their excess photosynthetic products. They have some different photosynthetic pigments. So they have chlorophyll A and B. So 2 different types of chlorophyll. And then they also have accessory pigments. So those would be carotenoids and phycobilins. And those are their accessory pigments that absorb different wavelengths of light that help them to do more photosynthesis. So they capture light. Here's your little summary of cyanobacteria doing photosynthesis. So they take carbon dioxide and water, use some light energy to create their sugars and oxygen. So these cyanobacteria have accessory pigments and can undergo what we call chromatic adaptation, which is basically a response of the bacteria to the intensity of light. So depending on how much light there is and the intensity of that light, the bacteria will change how much of these accessory pigments that they have in order to be able to capture the most light or to help shield them from too much sunlight. So they can, because these pigments will absorb the sunlight and act like a sunscreen for them. So if the sunlight is super intense, they'll produce more of certain kinds of pigments to help to absorb that excess sunlight and protect them. Or if there's not a lot of sunlight, they'll produce other pigments which will help produce more of other pigments which will help to capture more sunlight and allow them to do photosynthesis. So they undergo what's called chromatic adaptation, which is kind of awesome that one single-celled little thing can do this. So cyanobacteria can exist as single cells, or they can also exist in these dense mats that are held together by mucus. And when they form these dense mats that are held together by mucus, they can form these things that are called stromatolites, which they look like little coral-like mounds of bacteria, basically. And when they form these mats with mucus, a lot of times the mucus will capture things like sediment and dirt and stuff, and then those things will be cemented together by the mucus and they'll harden and become these hard, actual physical structures that we call stromatolites. And here's a picture of them. So these little black things right here, all over the place in this picture, those are stromatolites. So they're basically rock-like things that were formed by bacteria and the mucus that those bacteria produce. Mucus, if you think of how if you spit, okay, your spit is kind of more jelly-like, that's mucus. Other photosynthetic bacteria. You have bacteria that will do photosynthesis. So remember, photosynthesis just simply means using light to put something together with. And so you have certain kinds of bacteria that will do photosynthesis but not create oxygen because they're going to use different kinds of chemicals in order to be able to do that photosynthesis. So you have anaerobic green and purple sulfur and non-sulfur bacteria that will do photosynthesis but not produce oxygen. They have a different kind of pigment in order to do that. That's called a bacteriochlorophyll. And they don't produce oxygen. So the rest of that you don't need to know. So here's a summary of that process. So these kinds of things will take carbon dioxide and hydrogen sulfide. They use light energy to create carbohydrates and sulfate. So they still create sugars for their energy source, but rather than producing oxygen, they produce sulfate. You can also have chemosynthetic bacteria that do chemosynthesis. So they're still autotrophs, but instead of using light to create sugars, chemosynthetic bacteria use energy from different chemicals rather than light. So it's chemosynthetic from energy from chemicals. They use chemicals to synthesize things. So they would use things like ammonium, sulfides, okay, elemental sulfur, nitrates, iron, okay, to all help to do a process that creates sugar. Chemosynthesis is less efficient than photosynthesis. So things that do this, the chemosynthetic bacteria, grow much more slowly than our photosynthetic bacteria. And so they're actually found around hydrothermal vents and some shallower habitats where those materials that they need are in abundance. So where the sulfur and stuff is in abundance, you'll have chemosynthetic bacteria. And again, they're autotrophs. They're still making their own sugar source, but they're just using something else other than sunlight to do that. And so they still start off the food chains. So around hydrothermal vents, chemosynthetic bacteria are the bottom of the food chain. They are your autotrophs. Here's a picture to help you see that. So this represents a hydrothermal vent. So here's your bacteria, okay? And these guys use the chemicals in the water like carbon dioxide and hydrogen sulfide to do chemosynthesis and produce carbohydrates and sulfur. And these carbohydrates can be used by the other living things to support the food chain. Another kind of super important bacteria would be heterotrophic bacteria. Heterotrophic bacteria do not make their own food. So remember, heterotrophs, they get their nourishment from another source. So that means that they're going to be needing a sugar source from some other place. So heterotrophic bacteria are decomposers. So they get their energy from outside sources like dead things that have died. And so decomposers will actually excrete chemicals onto dead things. And then those chemicals digest the dead stuff. And then the decomposers slurp up all of those nutrients. So these heterotrophic bacteria that act as decomposers break things down, and that's actually how they get their energy source. And they're actually hugely important to the marine environment because they take all of those nutrients and stuff that are trapped inside of like these dead things. They recycle them and they return them back to the marine environment so that they can be used by primary producers again, and so the ecosystem can go on. So heterotrophic bacteria, very, very important. Heterotrophic bacteria are found everywhere. They attach to the surface of organic particles and digest things. And they'll actually typically secrete mucus to help them cement themselves onto those dead things and then secrete the exoenzymes, which help to digest the food that they're attached to. So heterotrophic bacteria, as they attach to different things, they actually help with 3 hugely important processes in the ocean. So those are consolidation, lithification, and sedimentation. Consolidation is when these bacteria attach to certain dead things. They secrete this mucus that helps them to attach to it. And as they do that, mucus is slightly sticky, so other things around it also stick to that that bacteria, and so you get a bunch of things that that start to stick together, and so you take all of these things and you consolidate them into one, so it helps with consolidation. Consolidation can lead to sedimentation, so as things start to stick together and they get bigger and bigger and bigger, eventually they get too heavy to be able to like float in the water column, and they fall to the bottom and become part of the sediment. So these bacteria aid in sedimentation, the settling of particles out of the water column. They can also be associated with lithification. So as these bacteria attach to other things, and they secrete these enzymes, they can actually change the pH of the water around them, which can cause chemical reactions to happen, and they can form minerals between particles, which is this process of lithification. So they can form like a cement between different particles. Sedimentation, so as those particles settle out, they can actually be in the form of what we call marine snow. So they're just basically large, like cobwebby, like mucousy drifting structures that are formed by lots of different plankton. And that's where these particles can accumulate, and marine snow is actually the main food source for the deep sea. We'll talk more about that later in the semester. So this is just kind of a summary of what bacteria do in the ocean. So you can have primary producers, so you can have chemosynthetic bacteria that will be using chemicals to make sugars, or you can have photosynthetic bacteria that will be doing photosynthesis using sunlight. Okay, but the photosynthesizers will be producing oxygen. When these guys get eaten by consumers, okay, that energy that they produce gets passed up the food chain. And then when these guys die or they release waste, the decomposers will break them down and release nutrients back into the water in order to supply nutrients for the photosynthesizers, and so the cycle can continue. So bacteria take place in pretty much all of the different steps of the food chain in the ecosystems in the ocean. Bacteria are also going to do this crazy important thing called nitrogen fixation. And nitrogen fixation is the process that takes molecular nitrogen and converts it into a usable form for phytoplankton. It takes nitrogen gas and makes it into the ammonium ion. And nitrogen in the form of ammonium is actually one of the major nutrients that phytoplankton need. So all the little microscopic plants that live in the ocean, they need this ammonium in order to be able to make things like proteins. So they are important. Basically, these bacteria that do this process of nitrogen fixation take nitrogen in an unusable form, nitrogen gas, and convert it to ammonia, which can be used by the primary producers. The kinds of things that will do this are cyanobacteria and a few archaea that have this enzyme that's called nitrogenase. And it takes the nitrogen gas and it is able to break that super strong bond between the two nitrogen molecules or atoms and convert it into ammonium. Typically, this process of taking the nitrogen atoms and splitting them and creating ammonium, it has to happen in an anaerobic environment, which means it has to happen where there's no oxygen, because the nitrogenase enzyme is sensitive to oxygen and will actually break down in the presence of oxygen. So nitrogen fixation typically occurs in this place in these bacteria that it's called a heterocyst. So it's a single cell that has a super, super thick cell wall, and it's basically where they actually alter photosynthesis so that it doesn't release oxygen, so that the nitrogenase can work and create nitrogen. So here's kind of what it looks like. And you actually look at this when you look at the cyanobacteria under the microscope for our lab this week. You'll see you get a bunch of like these little bacteria cells, all circles joined together, and then you'll see there'll be like one big cell, and then more little cells. This big cell, that is the heterocyst. That's the cell that has the giant cell wall to try and block out all of the oxygen that's floating in the water, that's trying to get in. This big, thick cell wall prevents the oxygen from getting in, and then any oxygen that does get in is quickly used up so that it doesn't affect the nitrogenase, which is doing the process of nitrogen fixation. And then any of that ammonium, that excess ammonium that leaks out of the cell, can then be used by other primary producers around there to do primary production and produce sugars for other living things. Nitrification is the process where you take ammonium and convert it into nitrite and nitrates. And then those are that's the form that you'll need all of your nitrogen in order to be used by the primary producers. So bacteria also do the process of nitrification. So here's your picture to help you see it. So you have dissolved nitrogen gas, okay, that gets absorbed by cyanobacteria, okay, which takes the two nitrogens, splits it, and converts it into ammonium. Okay, that ammonium can then go through nitrification, which converts into nitrites and then nitrates, which can then be used by phytoplankton. Okay, or the ammonium can go directly to the phytoplankton. So bacteria do this. So bacteria are primary producers. They do photosynthesis, create sugars, and start off food chains. They do the process of nitrogen fixation, which allows for other phytoplankton to exist, and they decompose things and recycle nutrients. So bacteria are crazy, crazy, crazy important to the ecosystems of the oceans. You can also have symbiotic bacteria. These are bacteria that will live inside of other things and do things for their host. So you've got chemosynthetic bacteria that will live in tube worms and clams and provide food for them. You also have some deep sea and nocturnal animals that have bacteria that are bioluminescent, meaning they create their own light. So you have certain animals that will have organs that are called photophores, where they will keep their bacteria, and then when they need them, they'll cause the bacteria to glow for various reasons, either to attract a mate, to attract a prey item, or to escape from predators. Archaea. Archaea are teeny, teeny, tiny little things smaller than bacteria. They're also prokaryotic. And these are what we call extremophiles. So archaea are like bacteria, but they can withstand more extreme environments. So they can withstand super, super salty places. They can withstand low pHs. They can withstand high pressures and high temperatures, and also low temperatures. So they can withstand a lot of different environments. They are a little bit different from bacteria. Here's how they're different. Their cell walls don't have the same like sugar amino acid compounds that the bacterial cell walls do. So their cell walls are different. And also their cell membranes are going to have different types of lipids, different types of fats. So that's going to help to stabilize them. So they have different things in their cell wall and their cell membrane, which helps and allows for them to actually live in these extreme environments. So archaea can be photosynthesizers, chemosynthesizers, or heterotrophs, but most of them are what we call methanogens. So they take organic matter and make methane. Methane's like the gas smell that you smell if you like drive by a cow farm. Okay, that's methane. Halobacteria are photosynthetic, and they trap light using these things that are called bacteriorhodopsins, which is a protein that is similar to the protein in your eye that captures light that allows you to see. Different kinds of archaea. You have hyperthermophiles. Okay, these are things that can withstand high temperatures. Hyperthermophiles. Hyper means high, thermo temperature, philes is like lover. So they are high temperature lovers. They're found in temperatures that exceed 100 degrees Celsius. That's hotter than boiling water. So they are able to live in those. Here's a couple examples. So strain 121 survived 2 hours of incubation at 130 degrees Celsius, which is really hot. It's over 200 degrees Fahrenheit. So it was able to withstand that kind of temperature. All right, eukarya. So these are types of things that have a nucleus. So all the DNA is contained in a nucleus. And examples of eukaryotes would be plants, animals, fungi, algae, and then some single-celled things that are called protozoa. And so we're going to talk about plants and animals and plankton, but right now we're going to discuss briefly fungi. So fungi are eukaryotes, and they have a cell wall made of chitin, which is a special kind of sugar. It's similar to, it is what you would find on like a crab shell. So crab shells are made out of chitin, and fungi have a cell wall that surrounds each of their cells with chitin. They're typically the ones that we find in the ocean, unicellular yeasts, so they're a single cell. You do have filamentous fungi, and they'll have these long filaments that are called hyphae, and then those hyphae can join together and form this thing called mycelium. And if you ever see like bread mold on your bread, what you actually see, like the black stuff on your bread, that is the mycelium. So here's a picture. Okay, so if you can kind of see it, there's a bunch of like little hair-like things. Those would be the hyphae, and then this right here would be the mycelium. Okay, and they're found, that's like grains of sand. So they're teeny, teeny, tiny little things. They are important in the ocean and in food chains because they're decomposers that help to recycle organic material. And one of the things that they can actually recycle that most other things can't is this thing called lignin, which is actually an important part of wood. So as wood and stuff gets washed into the ocean, the fungi are one of the things that's actually able to break it down. Fungi store their energy as glycogen, so kind of like it's called animal sugar. So they store their energy like animals do in the form of glycogen. There's 4 phyla of fungi, which you can see here. And the sac fungi, the Ascomycota, that's the most abundant in the ocean. So salinity is toxic to fungi. So how do they live in a place like the ocean? They're very hard. It's hard for them. Basically, they spend most of their energy devoted to maintaining their salt-water balance. And so they have a hard time. So there's not much of it that can survive there. And what you do find, most of it's found on wood that comes from land. But they can also live in grass and salt marshes or on other kinds of algae or sand. And again, they are decomposers, so they help to digest the remains of dead things. So they help to return nutrients to the primary producers, which is very important. Okay, so that concludes our extra lecture that you need for honors. You can read all of this in your book in chapter, let's see, goodness. Well, it's labeled marine microbes. So if you need to, you can read the section, and if you need to, come and see me for any questions that you have.